Oh, actually, I need to do on everything. Do you guys have any questions over what we did last time with regression? Questions about? All right. The reason we're going through regression is, once again, that regression is a method that is used in virtually all of the papers that you guys are going to be dealing with. So we had talked a little bit about what R squared means in terms of the spread of your data and the ability of the X variable to explain variation in the Y variable. And then we talked about these two really terrible kind of figures where, although the slopes are significantly different from zero, the explanatory power of the regression isn't very high because the R squared values are really, really low for both of these plots. 0.075 for this one, 0.09 for the top one. And so R squared is a good diagnostic statistic, but it's not a statistical test of whether the slope is significantly different from zero. And ultimately, that's what a regression is testing, whether or not the slope is significantly different from zero. And it can be significantly different from zero in a positive way or a negative way. In this case, these are all positive regressions. The other thing is, you don't always have to test whether the hypothesis is a slope of zero, whether the null hypothesis is a slope of zero. You can test any slope. You could test to see if the slope is different from a slope of 1, for example. And so you can specify what the slope is in the null hypothesis. And that's actually what the second Brody paper is going to be doing, is it's not testing against a null hypothesis of a slope of 0. It's testing against the null hypothesis of a slope of 1. And I'll explain why that's the case. Now remember that we have regression data that we've looked at before. In this case, you can look at offspring trait versus parent trait and use the slope of this relationship to measure heritability. Back in lab, we dealt with this shoe size data set where we had 408 data points on shoe size and height in men and women, and asked basically, do men and women have different shoe sizes? Normally, to answer a question like this, you would use something like an ANOVA, where you just look at shoe size. But because we know that the taller you are, the bigger your shoe size is, we have to also then account for the fact that males and females are generally different heights. And back when we were doing this in lab, I presented you with this figure where you basically had height on the x-axis, shoe size on the y-axis. The females are in red and the males are in blue. And so you can see that generally, there are no males down in these height ranges. There are no females in these height ranges for the most part. So partly the height that you are influences what your shoe size is, because these two things are correlated. Well, one of the ways you can remove the effect of height on these data is to basically measure what the residual is. How far are each of these sizes from the line predicted by this relationship? And so you can calculate the residual shoe size. If it's positive, you have a shoe size that is bigger than would be expected for your height. If you have a negative residual, it means that you have a lower than expected shoe size for your particular height. And then you can look at these residual shoe size values. How far are you above and below the line to basically correct for the influence of overall height in these data. And when you do that, what you find is that males still have bigger shoe sizes than females, but the magnitude of these differences is very different. When we look at the residual shoe size, it only varies from about minus 0.4 to plus 0.3, as opposed to here, there was a difference between about an 8 and a quarter shoe size to an 11 and a half shoe size, and that's a pretty big difference. And so these residuals, although they don't destroy the difference is much smaller because it's not confounded with the effect of overall height. And so the Brodies are actually going to use residuals in some of their work, and so it's important that you remember what residuals are. And just to go back, we talked about residuals with the P data back here. This is a hypothesized slope of 0 at an average weight loss for these flower beetles, and the residuals, once again, are just the differences between the hypothesized line and each of the individual data points. Remember that when you run a regression, what the regression does is it reduces the magnitude of these residuals. So to reduce these residuals, what you would do is you would actually have a line with a negative slope, and these residuals are all smaller. And so residual is just the distance between the actual data point and the line of best fit through the data points. So when we get to the Brody's data, they're going to be using residuals to look at the effect of body size on speed. But the other thing that they're going to be doing is they're going to be looking at these regressions of baseline speed, how fast the snakes were before they injected the snakes with tetrodotoxin versus post-injection speed. And they have two lines on here. One of these lines is the one-to-one line. The other line, the solid line, is the line that is the best fit through the data. And they're using this in the second paper that you're going to read. So the paper that you're reading for next Monday is going to have this figure in it. It's from Brody and Brody 1999. And what they're interested in is they're interested in whether or not there's an evolutionary trade-off or a physiological trade-off between resistance to tetrodotoxin and baseline speed. And how they're doing this is basically like this. If you have the speed that you're running, that you're slithering, before you get injected, and the speed after post-injection, what would this relationship look like if you were totally immune to tetrodotoxin? Matt?
It would be the same. So like if they started at 5, it would be at 5. If they started at 10, it would be at 10.
Okay, so the faster you are, baseline speed, if tetrodotoxin has no effect, you should have the same post-injection speed. And so what we would have is we would have a one-to-one line. The speed that you are before you get injected is the same as the speed that you are after you get injected. So you're exactly right. If you slither at 5 centimeters per second, then you would slither at 5. If you slither at 15, you would slither at 15. So there would be a one-to-one relationship, and this line would pass through the origin. Somebody else. If, on the other hand, you were affected by tetrodotoxin, but the effect was the same regardless of what your baseline speed was, what would happen to this line? If you were affected by tetrodotoxin, meaning that it slowed you down, but it didn't slow you down more or less depending on what your baseline speed is, what would happen to this line?
Would it just be a flat line?
Nope. If it affected everybody, but affected everybody equally.
Would it just have a lower slope?
If it had a lower slope, that would mean that it would go down, meaning that it affects fast individuals more than it affects slow individuals. So the effect of tetrodotoxin would be that it would slow everybody down, but it would slow everybody down to the same degree. So what would happen to the line? The slope would be the same, but what would change?
It wouldn't be 1 to 1 again.
Well, no, because then that would be a change in slope. What'd you say, Avery?
The intercept?
The intercept would be different. What would happen to the intercept?
Just move down.
It would move down. So this would be the effect of tetrodotoxin, but there's no trade-off. Some individuals aren't affected more by tetrodotoxin than others. Everybody is affected by tetrodotoxin the same way. They're all affected by tetrodotoxin to this degree. So it's still a one-to-one line, but the y-intercept has been shifted. If there's a trade-off, we would expect a change in slope. So if this is the effect of TTX with no trade-off, and we go and we gather a bunch of data on snakes, we could basically calculate the average baseline speed, calculate the average post-injection speed, and that difference in mean would indicate the magnitude of the trade-off. And so if there is no, the magnitude of the effect of TTX, but if there is no trade-off, we could just plot a one-to-one line through this mean right here. Notice that this dotted line is parallel to these lines, and this is just reducing the y-intercept to the degree to which tetrodotoxin is having an effect, because the tetrodotoxin effect is the degree to which baseline speed on average is different from post-injection speed on average. Is this dotted line a good fit through the data? Avery is shaking her head. No, why?
It's just not, there's too many like differences for like above and below.
Give it to me in terms of residuals. What's happening with the residuals?
There's too big a difference between the average one.
Yeah. If we compare this one-to-one line to the data points, residuals up here are all negative for the most part. Residuals down here are all positive. If we wanted to have a better fit through the data, what would we do? We would tilt what?
Like lower the slope.
Yeah, we would lower the slope. We would make the slope more shallow. It would bring this end of the line up. It would bring this end of the line down. And so this is actually the observed line going through the data. This is the best fit line. And this represents an effective tetrodotoxin and an effective trade-off of speed and tetrodotoxin resistance, such that fast snakes are slower on average than slow snakes are after they've been injected with tetrodotoxin. And so in this case, they're going to be testing not to see whether the slope of this solid line is different from a slope of 0. They're going to be testing whether this solid line is different from a slope of 1. And when you get rid of all of the rest of this stuff, it ends up looking like this, where the dotted line is the slope of 1, the solid line is the slope of the actual data, and they're interested in, is the slope of the solid line less than the slope of the one-to-one line? And in all of these cases, those solid line slopes, which are the data, are lower, indicating that faster snakes are more affected by tetrodotoxin than slower snakes are. And this is the crux of the second paper that you're going to be reading. So you can use regression for lots of different kinds of things. This is one of the applications of regression that they're going to be using in the papers that you read. The other one is going to be looking at residuals to remove things like the effect of size. Questions about regression in general? This is by far the most useful animation in this class in terms of helping you figure out what the effect of tetrodotoxin is, when there's not a trade-off, what the data would look like if there wasn't a trade-off, and then what the actual regression is, and then how that decomposes to look like these figures here, okay? All right. This PowerPoint is already up. It's been up since last week. This PowerPoint is not up because I'm going to ask you to be doing some things in class, and I don't want you to have the answers just yet. So I'll post this after we're done with class today. So we need to go back and revisit quantitative genetics because Butch Brodie Butch Brodie III is actually a quantitative geneticist, and when he refers to heritabilities and things like this, he's using quantitative genetics. So we want to revisit quantitative traits and what they are. We want to look at the evolutionary response to selection for a single trait. We've already done this. We did this back when we studied the finches in the Galapagos. So this is going to be a review. We're going to look at genetic correlations between traits. So we're going to look at multivariate evolution, because in this case, we're interested in not only resistance of snakes to tetrodotoxin, but also we're interested in things like second variable is. We've got resistance to snakes, but also what their baseline speed is. So are these two traits being determined by different genes, or are they actually being determined by the same gene? So is resistance being traded off with baseline speed? And so we can calculate the evolutionary response to selection for genetically correlated traits using the same methods that we used in single trait variables, except the equation is slightly more complicated. And then we can explore how these genetic correlations between traits can alter the expected path of evolution. So just to review, quantitative genetics is the study of these continuously varying traits. We use it to study quantitative traits, things like height and sprint speed and resistance. The math is once again rooted in simple Mendelian genetics, and our goal is to predict how selection acts in the next generation. So these things are controlled by multiple genes. They're polygenic traits. They occur in groupings called quantitative trait loci. Each dominant allele adds to the effect of the genes on the phenotype, and so ear length and corn, crop yield, fat content, and meat height, all of these things are quantitative traits. This is all review. We talked about doing a trihybrid cross where the recessive alleles add nothing and the dominant alleles add 1 unit to whatever. And when you do a Punnett square of these heterozygotes being crossed with one another, remember this Punnett square from hell, everyone? This is what it looks like if you look at the genotypes. But do genotypes matter when we're thinking of quantitative traits? What matters?
Phenotype?
Hmm?
The phenotype?
Well, what determines phenotype in terms of how is genotype related to phenotype? What matters most? Yeah, then just the number of dominant alleles. So we can take this complicated thing, bust it down to the number of dominant alleles. You see we have 6 up here, we have 5, going down to 4, going down to 3, going down to 2, going down to 1, down to 0. Here at the end, we talked about the symmetry of this and how the symmetry of this Punnett square is reminiscent of the symmetry in a dihybrid cross. And we showed that what this results in is these normal distributions of polygenic traits, from 0 number of contributing alleles to 6 contributing alleles. And the most abundant phenotypic class is the phenotypic class with an average number of alleles. These distributions can then be used to look at evolution because these distributions change over time under selection. Who can remind me what the Breeder's equation looks like? This is second test material. Any takers on Breeder's equation?
Isn't it something heritability squared?
Heritability is not squared, it's just h squared is the
it's
shorthand for it. So you have it? Sounds like you have it.
Do I do it over here or over there?
Do it over here, since I started.
Isn't this heritability in the S?
Does what? And those are multiplied, added together, subtracted from one another?
Multiply?
Okay, they're multiplied together, and what will they predict? Somebody help her?
What?
Delta Z.
Okay, so what is delta z? Somebody, who said delta z? Actually, delta z bar. When we put a bar over something, it's mean what?
The sum of the,
It's not a sum of anything.
oh,
Change in what?
Average.
Average what? Emily? this is not, we're not talking statistics.
Okay.
Change in average.
Bless you.
What is S?
It's the selection.
So it's the strength of selection. What is H squared? This is heritability. So selection on what?
Strength of variability.
Strength of selection on what?
The evolution of the species.
But on what in particular about that species?
Trade.
Okay, so a trait, so a phenotype. Selection on phenotype. So if phenotype is what's being selected on, what's changing? Average phenotype, yeah. So if you have a phenotype that is determined genetically, selection is acting on it, the heritability times the selection tells you how the average phenotype changes over time. That delta Z is also referred to as the evolutionary response to selection, but the thing that is responding to selection is the phenotype. So whatever phenotype it is that you're interested in. And so this is the equation, but in this case we're only looking at one phenotype Z, whatever it is. That should be Z bar because it's average phenotype. I don't know what happened to the bars. So the trick though is how do we do this when we have two traits that we're interested in? So in the case of Butch Brodie III, he was working on his dissertation work in Southern Oregon, and he was looking at Thamnophis organoides, one of the garter snakes there. Remember that this is the slug and worm specialist. And he was interested in how stripedness versus being checkered, being striped versus being checkered, was related to behavioral traits. And in this case, he was interested in the behavioral trait of reversals. How frequently do you change direction as you're running away from the predator? And in this case, the predator is actually him chasing a snake around a circular racetrack with paintbrush. Paintbrushes are usually what we use to chase animals down racetracks with. And so he was measuring speed, and he was also measuring endurance. And when you measure speed, you just run an animal down a linear racetrack. It has brake beams, and it records how quickly you go from one brake beam to another brake beam. It calculates speed for you. But he was also interested in endurance, and so he also built a circular racetrack. He would crawl underneath it, pop up in the middle of it, and he would chase snakes around in a circle, and he knew what the diameter of the circle was, so he could look and see how many times the snake went around the circle before it finally just tuckered itself out, and that would be a measure of endurance, how far they ran. But he ran into, so what happens with reptiles, because they're cold-blooded, is they actually go into anaerobic respiration really quickly. So reptiles aren't generally known for having a lot of endurance. And oftentimes we think about speed and endurance being negatively correlated with one another. Sprinters make terrible distance runners make terrible sprinters. As it turns out, there's not a lot of truth to that. But that's a story for another time. But when he was chasing these snakes around in these circular racetracks, he noticed that they had a tendency to turn around on themselves. They'd be going in one direction, and then they would just flip around and go the other direction, which actually made it more difficult to measure endurance because you had to account for those changes in direction. But he also started to notice that not all snakes reverse themselves to the same degree, so he started recording that as well. And what he found was that these two traits, how stripy you are versus how checkered you are, is also related to how often you tend to reverse yourself. So we can look at something like stripedness, and let's just assume that this is stripedness. And this is offspring stripedness, and this is female adult stripedness. How does a female stripedness relate to the offspring stripedness? And he created an index that would allow you to quantify how stripy you are. What is this slope telling you? Because this is offspring trait versus female trait. This is telling you heritability. So heritability for this trait is 0.707. This is that relationship. If selection acts on females such that selection is pushing the population towards being stripier, then we should expect a response to that selection to appear in the offspring that is represented by this change in average phenotype of the offspring. This is the breeder's equation. The change in offspring phenotype is due to selection multiplied times this relationship showing heritability. We can take this and work the math. We can see what's the difference between the populations post-selection versus pre-selection. That's this big of a change times that heritability. That's how much you would expect this population to change. Doesn't matter how you work it out, it always ends up being the same. We did these kinds of equations back in the day. Same thing with reversals. If we have this relationship between reversals, we see that there's actually a lower heritability in this case. And in this case, perhaps reversals is being selected against negatively, such that selection is pushing individuals to reverse themselves fewer times. And if that's the case, then we should expect a decline in a tendency of snakes to reverse themselves. So in this case, post-selection minus pre-selection means gives you a negative selection differential, multiply times the heritability, and so you should expect reversals to change by this much from one generation to the next. And this is basically directional selection acting, but in this case, directional selection is acting on two traits simultaneously. Now here's the tricky thing. We can look at the evolution of stripedness by itself, we can look at the evolution of reversals by itself, but it might be more productive to look at the two evolving in tandem, especially if there's a reason to think that they might be genetically related to one another, these two traits. And there are a number of ways that two traits can evolve in tandem with one another. One way is that you have two traits that are both controlled by the same genes. What is that called? When one gene controls more than one trait. What?
The complex gene?
Nope. It's a genetic term. I'm certain you learned this in high school. Single gene controlling multiple traits. Don't move your head yet. What?
Phenotypic?
Nope.
P.
Hmm?
P.
L.
N? Sadness. Hmm?
Pleiotrope?
Pleiotrope. Pleiotrope. Oh, I spelled it wrong. Goodness.
Well, that's why we couldn't guess it.
Pleiotrope. That's why you couldn't guess it, of course. Pleiotropy. Pleiotropy is when you have a single gene that controls a more than one trait. And this is known to happen in lots of cases. Growth hormone, the gene that codes for growth hormone, controls lots of different traits in your phenotype because growth hormone has these generalized sorts of effects on your development. And so, if you have more growth hormone, it affects your height, it affects your shoe size, it affects all sorts of things about you. So that's one way that you can get two traits being correlated with one another. Another way you can get these traits being correlated with one another is if the genes lie next to one another in close proximity within the genome. And so they always tend to get inherited together because they don't undergo the process of disjunction during the formation of gametes. Because they lie on the same chromosome in close proximity to one another, they tend to get inherited together, and so they tend to be correlated with one another. So that if selection is acting on one of those genes, it tends to drag the other gene along with it. So there are multiple ways that you can get traits that are correlated with one another, and in this case, he was interested in whether these traits were correlated. So what they did, this should all be familiar to you guys now, collected a bunch of snakes, brought them back to the university, and raced them down these linear racetracks to measure speed, and also raced them around circular racetracks. I couldn't find a picture of the circular racetrack. And then they scored them as to whether they were more stripy or more checkery, and recorded their reversals. And in the papers that you read, it also includes speed as a character, but they don't talk about it very much. Brady doesn't talk about it very much. So, what this does when these selection acts on one trait and it also affects other traits is it acts through genetic covariances, and covariance is just another way of expressing correlation when two things are related to one another. Well, how do we determine if traits are genetically correlated? We talked about this briefly when we were talking about the finches. To measure heritability, what do we run a regression on? What's on the y-axis? And what's on the x-axis?
Offspring is on the Y and females on the X.
Okay, so parent trait is on the x-axis. So this is what it would be. Mid-parent phenotype on the x-axis, average offspring phenotype on the y-axis. This is how we measure heritability of a single trait. How would we measure genetic correlation or covariance? Using these same kinds of methods. Let's say this is stripe and this is stripe. How would we measure the genetic correlation between stripe and reversal? So this would be checkered versus striped. This would be then heritability.
Would you add another axis?
No, we wouldn't add another axis. Thank you for playing.
If the reversals are generally the same as the stripiness that they get from the parent, then
Okay, but how would we
we measure the parent's reversals and stripiness and measure the baby's.
okay, but in terms of getting, because we can also use this slope to also give us the genetic covariance. But only if we do what with this regression? We now want to know how stripe is related to reversals, right? What would we change? This basically asks, how does the parental phenotype for stripe affect the offspring phenotype for stripe?
So would you do stripe and then reversal on the Y?
Put trait 1 on one axis and trait 2 on the other axis. And this gives us a correlation, but now it's not a correlation between parents and offspring for a single trait, it's a correlation between parents and offspring for 2 traits. Reversals on one, stripe on the other, or vice versa. It doesn't really matter, and actually you would do both of them in this case. And when you get that, you can see that these might be positively correlated with one another. You might find that they're negatively correlated with one another, or you might find that they're not correlated with one another at all. But now, when we look at the breeder's equation, that only has room for one trait in it. And we need to have another way of expressing the breeder's equation, but in this case, it expands that breeder's equation out to multivariate traits. And this was, the first paper that I read about this was Landy and Arnold in 1984, the year I graduated from high school. And what Landy and Arnold basically did was expanded this breeder's equation that was proposed back in the 1920s to include multiple traits. And in this case, we now have 2 traits that we're worried about. And this goes into a vector of traits. And now we have a matrix that tells us about the genetics. We have the heritability, or the additive genetic variance of trait 1, whatever trait 1 happens to be. We have the additive genetic variance of trait 2. And then in these quadrants, we have the genetic covariances, how trait 1 is related to trait 2. We would multiply that times, in this case now they're called selection gradients, the selection acting on trait 1 and the selection acting on trait 2. And so this becomes a little bit of matrix algebra, but matrix algebra isn't really very hard. If you want to know how trait 1 is going to change, you take the heritability of trait 1, or genetic variance of trait 1, multiply it times the selection acting directly on trait 1. So that's G11 times the selection gradient. This could easily, just as easily be written if this was the heritability for trait 1 and that was selection of trait 1. But we have more than this going on. We also have selection acting on trait 2, and as selection acts on trait 2, it can influence trait 1 if this genetic correlation, genetic covariance, is non-zero. And so we have to add to that whatever the selection on trait 2 is that is acting on the trait 1 because trait 1 is correlated with trait 2. So this represents kind of the breeder's equation in the single case. This represents the breeder's equation when you allow genetic correlations to exist. Selection acting on trait 2 can influence trait 1 because they're genetically correlated. In the same way, we can calculate the effect of selection acting on the phenotype because of direct selection acting on trait 2, but we also have to consider the effect of selection acting on trait 1 affecting trait 2 through this genetic correlation. And so this whole thing basically breaks down to this equation and this equation. And then we can see how each of these things are evolving due to direct selection acting on the trait and indirect selection acting on the other trait. This is what I just put up on the board for you. This thing is referred to as the genetic variance covariance matrix. This is a vector of selection gradients, and this is a vector of the evolutionary responses to selection. You could express it in R's and C's if you wanted to. It doesn't really matter. We're going to end up using R's and S's for reversals and stripes here in a little while, where we have, oh yeah, there it is, reversals and stripe. R is additive genetic variance for reversals, S is additive genetic for stripedness, and the SR and the RS are the genetic covariances that relate stripedness to reversal. So we could go through and measure this is now, what are we measuring here? Is this a covariance or a heritability? Heritability, because we have reversals on the X and reversals on the Y. So 0.2451, this is for reversals, I'm sorry, this is for stripedness. Where does this go in this matrix? It's stripedness.
Oh.
It would go right there. This is for, well, okay.
Yeah.
Oh, sorry, I got it wrong. This would be GRR, because this is reversals, so it would go right there. This is stripedness, so 0.707 would go right there, and then whatever the genetic correlation is would go there, and they're the same, because they're always the same. The relationship between reversal and stripe is the same as the relationship between stripe and reversal. So then we would have, in this case, there's a negative relationship between reversals and stripe, and I don't know why there's not an equation on here, but this relationship would then go here and here. And once we know that, if we have a way of measuring selection on trait 1 and selection on trait 2, basically looking at how female reversals influence offspring reversals, that would be this difference between pre-selection and post-selection mean is what V1 is. Same thing with stripedness, the difference between pre-selection and post-selection would be what beta 2 is. If we then know what the genetic variances and the genetic covariances are, we know what the selection strengths are, then we can predict what the change from one generation to the other will be for trait 1 and trait 2, whatever trait 1 and trait 2 happen to be. And so here's a color-coded version of it. This is the effect on trait 1. This is the genetics of trait 1. Additive genetic variance, genetic covariance. This is additive genetic variance of trait 2 and the genetic covariance of trait 2 with trait 1. This gets multiplied times that gets multiplied times that, this gets multiplied times that gets multiplied times that. Direct effect of selection acting directly on the heritability of the trait. Indirect selection acting on the other trait, dragging trait 1 along through the genetic covariance. Direct selection acting on trait 2, indirect selection acting on trait 2 through the correlation of trait 1 on trait 2. This is what the expansion looks like. This expands out to be that. Questions? This is what all that looks like in words. This is the change in the mean of trait 1 due to selection acting directly on trait 1. Change in mean of trait 1 due to selection acting on trait 2. Mean of trait 2 changing due to selection on trait 1. Mean of trait 2 due to selection acting directly on trait 2. It's basically everything that I just said, except in words. We can change all of this to reversals and stripedness. Selection acting on reversals, affecting reversals directly. Selection acting on stripe affects reversals through the genetic correlation between reversal and stripe. Selection on reversal affecting stripe because of the correlation, and direct selection of stripe on the heritability of stripe to affect stripe. And that's what the whole thing, again, just in words, and this is what the equation looks like when you write it all out, as I've written it out over there, except that Trait 1 in this case is reversals. Trait 2 in this case is stripedness. So, how does this work? Well, we can look at reversals and stripedness, few reversals versus many reversals, checkered snakes versus striped snakes, and we can see how these things move around in phenotypic space. So go to iTunes U, go to this week's materials, and you're going to see a document labeled multivariate selection scenarios. Pull that up. And what I've basically done is I've given you numbers for the additive genetic variances and numbers for the genetic covariances. And I've also given you the strength of selection for each of these. And there are 4 different scenarios. And what I want you to do is, all your group members have abandoned you today, haven't they?
Yes.
Oh, no. Wait. Oh, yeah. That's your group. Come and join these guys. Work in your groups. Wait a minute. Who's in your group? Why don't you guys join those guys? And all of you are here today. So you guys stay together. So you guys join them. What?
I said we should get a book of that.
You go over there. You guys come over here. Figure out what the evolutionary response to selection under each of those 4 scenarios will be, and then we're going to show how they would graph onto a figure. So work on that for a little while, and put your heads together. You don't have to do it separately and then compare. You can help yourselves through this if you wish. No.
Oh,
You said groups, so.
Oh yeah, you are in that group. Okay.
So do you want us to just go over there?
Just go over there,
Okay.
yeah. Sorry, I
I was.
was looking at spatial distribution, not looking at names on the board. Yeah, go join those 2 guys. So you just have more heads to put towards the task. You have to answer these questions before you can ask other questions. Yeah, so start on page 1, work out page 1, because you need that in order to answer questions on page 2. And we're actually going to go through the questions on page 2 as a group. Okay. Yeah, yep. Order of operations. Even works for matrix algebra. Matrix algebra isn't very handy in many instances, but it's ultimately handy here. Has everyone made it through scenario 1? Alright, stop for just a minute. We're not going to have time to go through all 4 of them today. Work the other scenarios that you haven't finished before you come into class on Wednesday, because we'll continue working on this. Let me just show you how this works with scenario 1 so that it'll hopefully help you with the other scenarios. So here we have the base equation. These are the selection differentials, the strength of selection on reversals, which is really weak. The strength of selection on strike is much stronger. Notice how both of these selection gradients are positive. So selection is pushing both traits in a positive direction. So if we just look at the evolution of each of these separately, what should we expect to see happening in strike and happening in reversal? What's the characteristics of the snakes we should expect to see? Okay, so snakes should become stripier or more checkery? Stripier, and should they reverse themselves more or less? They should reverse themselves more, because this is positive selection acting on both of those traits. But you'll see that we have a negative genetic correlation, so that as selection acts on one, it will push the other one the opposite direction. So based on that, what would you predict to be the outcome of this selection acting on trait 2 and this selection acting on trait 1? Because there's strong selection here and weaker selection here, selection here is going to affect trait 2 heavily, which is stripe, but it's also going to affect trait 1 positively. And notice how the genetic correlation here is much higher than the heritability is for reversals. What would you expect the outcome to be of combining this genetic variance covariance matrix with this selection vector? Strong selection with strong heritability, but also a strong negative genetic correlation, you should expect this to change in what direction, positive or negative? This genetic correlation, though, is linked to this selection. Weak selection on a strong genetic correlation, strong selection on a strong heritability. You would expect this to be positive, but here you have weak selection acting on weak heritability, strong selection acting on a strongly negative genetic correlation. What would you expect this to be? Negative. And when you do the math, what do you find? You find that even though selection is pushing reversals to increase, what happens actually is reversals decline. Stripe is increasing, but it's not increasing at the rate that it would if we just had selection acting on additive genetic variance by itself. This would be quite high. It would be 0.07. something as opposed to 0.06. So this negative genetic correlation drags the overall change in the trait value down. What this looks like in trait space is if you start here, direct selection acting on reversals pushes reversals a little ways because selection is really weak. Direct selection acting on stripe moves stripe quite a lot because you have a lot of heritability and you have strong selection. Notice how both of these are in positive directions. This is the effect of direct selection acting on each of those traits. But when we look at indirect selection affecting these traits, we end up with selection on stripe dragging reversals backwards because there's strong selection on stripe and there's a strong negative genetic correlation. And when we look at selection of reversals affecting stripe, it also brings it down because, once again, of this strong negative genetic correlation, such that the overall change in the traits is that reversals go down and stripes go up because of the structure of this, in spite of the fact that selection on both these traits was positive. And that's why people developed methods for looking at multivariate evolution, because if you looked at these traits by themselves and just looked at selection, you would expect stripe to be increasing and you would expect the stripe to be increasing and you would expect reversals to be increasing. But because of this negative genetic correlation, as one goes up, the other one goes down in terms of trait value. We'll work on the rest of these scenarios on Wednesday. Work them out before you come, and we're going to do the same types of exercises. Do you guys have any questions about your lab reports that are due this week in lab? As you write your lab report, do you find the oral presentation draft that you made a couple of weeks ago? Do you find yourself needing to go back and alter that as you get a better handle on what you're doing because you're writing an introduction? Okay, because guess what happens next week? You make oral presentations, don't you? In lab. So, as soon as you turn that written report in tomorrow, you have digital copies of it. The first thing you should do is what? Go to your oral presentation draft and say, okay, now that we've been through a lab write-up, a lab report talking about what we did, how does this change what we're going to say and how we're going to say it in our oral presentation? Those things should be tightly related to one another as you move forward. Bring 3 hard copies of your paper into class this week in lab. We're not just doing this to run up your printing budget, basically depleting your printing budget. We actually have specific goals for each of those 3 copies. Print them and bring them. We're going to be using them in lab tomorrow and on Thursday. If you had a method or a set of questions that was, for whatever reason, not addressed in the process of you doing your work because things didn't go right or things didn't happen the way you thought, don't put those methods in your report because you don't report on the results of those methods, so there's no reason for them to be in the methods section if that applies to you. I know it applies to one of my groups in the Tuesday lab. It might apply to the groups in the Thursday lab, I'm just not sure. Roberta.
So like when we want to talk about like, how you said things that we didn't implement, but like that we'd like to add.
You can throw those things into the discussion. So there are things that you guys wanted to measure that you weren't able to measure because you actually did too good of a job of retarding growth and things like that. Those are all things that can go into your discussions as to what you can do in the future. Yep, certainly. But don't put in methods about the Lightform machine.
Yeah.
Don't put in methods about cross collecting because you guys didn't. You got my email that you basically said you guys didn't poop very much.
I just didn't know.
Yep. All right. I'll see some of you tomorrow. I'll see the rest of you on Wednesday.